Thank you for joining us tonight for our career conversations. And tonight we have Brianne Rose Conlon, from a graduate from 2015, who had the wonderful experience of working with Professor Lubkowitz when she was an undergraduate, and worked on his maize research, and was funded through his NSF grant to do some research in Florida for a summer, I guess it was, right? And she sort of, I guess, got hooked on maize, or at least found it quite interesting, because for the last few years, she's been working in a maize research lab at Cornell. And tonight she's going to tell us about her path to biological research and what a day in the life of a laboratory research technician is like. So I present Brianne Rose Conlon. Thank you, Brianne, for being here tonight.
Thank you. And I'm going to share some slides that I put together just to guide me as I talk about my experience. Ooh, that's not what I wanted to do. All right, so thank you for that introduction. And like Professor Talentino said, I am a lab technician in a plant developmental biology lab. This is actually my boss right here, Mike Scanlon. So it's his lab. These are some of the previous lab mates. Can I get a better laser pointer? So that's up. Okay. Yes. Okay. So the outline of some of the things that I'm going to talk about tonight is, first I'll start with my life at St. Mike's, and then the time I spent right after graduating from St. Mike's, and how that brought me to becoming a lab technician and what it means to be a lab technician, and then where one might go if they decide to try out being a lab technician and try out research. All right, so when I was at St. Mike's, so I was there from 2011 to 2015, and I came in undecided. That's going to be a common theme as I talk about how I got to where I am today, is I haven't really had any set direction or big overarching goals. I didn't come into college knowing I wanted to become a nurse or something like that. And so I think it's good to mention that, like, that's totally fine. And I have been able to be successful thus far. And so if you're still trying to figure out what you might want to do, that's totally fine. I am still trying to figure out what I want to do. But what I ended up deciding to major in was biology. And then I also had a chemistry and a math minor. And some of the key courses that I took that have really been applicable to my life in research right now. So I took molecular genetics, and I also took a plant biology course. Those were both with Professor Lubkowitz, actually. And those are really, like, very applicable to what I'm doing right now, because the lab is a developmental biology lab. We look at genetics on the molecular level. Those have been super applicable. I also had a chance to take computer science, and I would definitely recommend taking some of those intro computer science courses if you're thinking about getting into an academic setting or a research setting, just because it's really nice to have that background, because a lot of the, like, data analysis and things like that, the more sophisticated you get, the nicer it is to have a background just understanding coding and things like that. I didn't take cell bio because, like I said, once I decided to be a bio major, I still didn't know exactly what I might want to do with that. So I took a very broad breadth of classes, and cell bio would have been helpful in this lab research setting, but it just meant that I had to do some catch-up work on the job. I also was able to study abroad in college, which was really nice. It was, I think a lot of programs, it is possible to get at least 1 or 2 courses that can count towards your major, and can give you some more experience in things outside of what St. Mike's might be able to offer if you're at a larger university, which might be nice to see some different things. In terms of things that I did over the summer while I was in college, I did, as Professor Talentino mentioned, spend 1 summer in a maize genetics lab down in the University of Florida. And I found that, or got that job through Professor Lubkowitz. And that obviously directly relates to what I'm doing now. And I do think that if you think you might want to go into research, trying to get either a summer position or even TAing a class or something like that would be a good opportunity before graduating. I also put down some of the things I like to do for fun in college, ultimate frisbee, and I had a radio show. And I have found that with like networking and interviewing for places and just making connections, it has been very important that I have things that I was passionate about, things that I was really engaged in while I was in college, even if it's not particular things that might be like part of your professional self, you can still make connections with people on the things that are broader outside of your professional self, and it's helped me make connections. I also wanted to mention, I put down making authentic connections with professors, staff, and peers. So if you think that you want to go into an academic setting, the whole networking is, I found to be super important, because higher ed and academia is a pretty small world and everyone knows each other from university to university. Once you get into a field, everyone knows each other and they go to the same conferences and, you know, like they're up on each other's lives. They probably were in a lab together at one point in their career. So making those connections has been really helpful for finding jobs, because on that same note, they know each other and they also are all super busy. And so with hiring and things like that, having that connection and having someone that they know who can vouch for you is really important. So now we'll move into once I graduated and what I did for the first 1.5 years after graduating. And so it took me 1.5 years to get to Cornell. And I was all over the place. So, you know, I graduated from St. Mike's, decided I wanted to take some time off. So I worked at a summer camp in Washington. And then my initial plan was to move back home afterwards. So that's where number 3 is. Move back home and apply for jobs and hopefully find something where I get to use my degree, you know, what the goal is of going to college. But once I moved back home, I quickly realized that looking for a job when you have no responsibilities and you live at home is not a fun time. And so quickly abandoned that. And actually talked to professors at St. Mike's who helped advise me during that time, because I just needed some help realizing that it's okay to just abandon ship and do something fun, which it totally is. And so I ended up moving to Utah for a winter to be a ski bum. I worked in a ski shop and I skied a lot. And after that, I moved back to Burlington. I worked at a cheese shop. Again, just odd jobs. doing what I had to do before actually landing this job here where I am doing biology, which is what I studied. So that was the goal of college. So then how did I get this job? Like I said, I decided to move to Park City and pursue something fun. But when I did that, I set a goal for myself that I had to line up something for after Park City that was using my degree, because even if I didn't know what I wanted to do, I wasn't going to figure it out if I wasn't in the field itself. So I knew I needed something in the general biology realm. And the first thing that I did when I started looking for jobs was I reached out to professors that I had worked with or had in class and just asked them, have you heard of anything? Basically, like, hi, how are you doing? Do you know anyone hiring? Just throwing it out into the void. And it just so happened that I contacted Professor Lubkowitz again, common theme, but the week that he was at a conference. And so he's at a conference, he now knows that someone is looking for a job, and he was able to put me in contact with Mike Scanlon, who is the lab that I'm now working in. And so I think once I was put into contact with Mike, the things that ended up being really important to him when he decided to hire me were that I did have some form of previous lab experience. So I wasn't completely new. I at least understood something about being in a lab, even though I didn't have extensive research experience, I had something. And then the letters of recommendation were super important. I think that's basically how he decided that he was going to hire me, was that his peers or his colleagues recommended me. And so that was enough for him to say, okay, you are worth hiring. And I wanted to throw in here, just so people have an idea of what a lab technician might make. When I was initially hired, I was hired at $20 an hour, and that was for a full-time job. Okay, so then what is it actually like on the day-to-day of what I do? So one of the fun things about being a lab technician working in research is that every day is different. And there's a lot of freedom in what I do. And so some of the different things that I might be doing on any given day would be planning experiments, which might involve reading primary literature, reviewing protocols, things like that, or just thinking about where our project's at and where it needs to go. I also obviously spend a good amount of time in the lab running experiments. As a tech, one side note with running experiments is it's not, you don't just get to run the experiments you want to run. You are an employee of the lab. And often if there's some type of really basic or menial task or data collection that has to be done, it might fall on you. Like I spent a month in basically a closet taking pictures on a microscope once, but you know, like it's okay.
It's a new skill. It's a great new skill.
Yeah. Also, if it's a biology lab, you'll probably be maintaining some different stocks. We have plants. We also have seed stocks. If it's a cell lab, you might have cell lines that you have to take care of, things like that. There's a bunch of lab maintenance that I'm in charge of because I run the fine line between lab technician and lab manager. So like if any equipment breaks down, I might need to get it fixed, or there's some administrative things that I might be talking to administrators, things like that. And then meetings and seminars, like we have weekly lab meetings. There's weekly plant biology section seminars that I like to attend, usually things throughout the week to go to. it'll be any combination of these different things that I'm doing in any given day. And then I wanted to give a really brief outline of a recent publication that I was able to work on, just because I felt like coming from a smaller school, it's hard to fully understand. I didn't really understand what research was, I would say, coming from a small teaching school and then going to a large research university. I just want to highlight some of the different, like from a journal article that comes out, my name is on it, but like where might I have had little bits where I might have been able to participate in the research. So on this project, these are all of the other people whose names are on it. Phil was the postdoc in the lab who, this was his project. So he was the main person investigating this. And then Robin and Renna also, we used some of their older data, as well as Robin did some new in situs that were included. Okay, so I'm gonna do obviously some research talk, but I'm gonna try to keep it basic, meaning I'm gonna skip a lot of details. And if you have questions, you can obviously ask me about it, but I don't expect you to actually want to ask too many questions about the research itself. But so the gene that we were interested in for this study is narrow sheath, which Mike has been working on for a very long time. But so narrow sheath encodes a transcription factor, which is necessary for leaf margin development. So if you look at these images over here, you have a wild type maize leaf, nice and wide, as you would expect a leaf to be. And when you knock out narrow sheath, so when this transcription factor is not functioning, there's no margin on the leaf. That's just completely absent. You have this very skinny, narrow looking thing. So it's necessary for leaf margins to develop. And then on the right side here, these images are if you take a maize seedling and you cut it right at the base, what you'll see is this circle of, it's called the shoot apical meristem. It's like stem cell tissue in the plant, and it's surrounded by the youngest leaves circling it. So these are two leaves wrapping around the meristem. And what you see here is that narrow sheath is localized. So this is an in situ, so it marks gene expression, it marks the RNA. So there's gene expression at the very tips of a wild type maize leaf. And then on the right is an immunohistolocalization, and so that shows the protein. And so the protein is also at the very tips of these very young leaves, which makes sense, right? Because that's when the margin is growing. So it makes sense that they're localized right at the tips. And so in this study, oops, that didn't get fixed. We were looking at, we wanted to know what are the targets that are bound by the transcription factor and then modulated, meaning their expression either goes up or down, the targets of narrow sheath, so the targets of that transcription factor. And so a cool technique that I actually got to do, so we did laser microdissection RNA sequencing of the leaf. So on these pictures here, that's the same as those slices that I showed you on the last slide. I got to use this microscope that we have that has laser and just cut the tissue right out of the tips of the leaves for the wild type and for narrow sheath. And then we extracted RNA from that tissue and then compared what genes are being expressed in both the wild type and the narrow sheath. And when we look at the differences between those genes that are expressed, we see which genes are either up or down when narrow sheath is not functioning. So that actually, the bound and modulated should be opposite. Sorry, I didn't. So we did that to find the modulated, and then this should say bound. Phil did a chromatin immunoprecipitation sequencing assay called ChIP-seq, which basically you have the transcription factor it binds in the genome, and you can fix that so that it sticks together and won't come apart. And then you break the DNA up into lots of little pieces. You sequence the DNA that's attached to your specific transcription factor, so to narrow sheath, and it tells you where in the genome the transcription factor was binding based on the sequence. Like these peaks here are where you had high abundance of this binding. So that gave us the bound. We had the modulated from the RNA-seq. And when we combined those two data sets, we had 55 target genes, which were in both data sets that were candidates for targets of narrow sheath. And then, so to look at which of these targets might be actually functioning in the plant, we moved on to arabidopsis, which is like it's the model organism for plants. It's like the lab rat of plants. And I did a lot of the arabidopsis work, which was neat because I have never worked with arabidopsis, but it is like once you get into plants, that's what a lot of the basic research is done in. So it was cool to get to add this skill to my repertoire. So this is an arabidopsis plant. This is PRS, which is the ortholog of narrow sheath. So it's the arabidopsis ortholog. And one of the genes that was on our list of 55 bound and modulated was this gene ARF2. Again, the specifics aren't that important, but ARF2 is a known growth repressor in plants. So yeah, when ARF2 is knocked out, you get a larger leaf, meaning that when it's functioning, ARF2 is responsible for minimizing that larger leaf. So we wanted to knock out ARF2 in arabidopsis. And so we used a CRISPR-Cas9 construct to target the ARF2 gene and transformed that into arabidopsis to knock it up. And so I was able to get lines of arabidopsis where ARF2 was knocked out. And then we did some phenotyping of the arabidopsis after that. So in arabidopsis, it's a little bit different than in maize. So maize, you get the really narrow leaf from knocking out this gene. And in arabidopsis, you just lose the stipule. So if you look, or if we zoom in at this box on this leaf that's coming out of the stem, at the attachment point, there is this very small little growth that's a stipule. And when you knock out the narrow sheath gene, so PRS, you don't have a stipule anymore. And these are scanning electron micrographs. And this was another part of the project where I actually got to do this. So there's a cryo, so a freezing SEM on campus that I was able to go to and then take all of these images. So yeah, sorry, wild type, you have stipules. PRS, you don't have stipules. And then when you knock out ARF2 in the PRS background, you have stipules again. So this was some of the supporting evidence that led us to say that narrow sheath promotes growth at the margins, so stipules being analogous to margins, by repressing the growth effect. So that's just a small part of the bigger story that we published, but I thought it was nice to showcase like different steps along the way, and as a technician, I was able to help at certain aspects. So switching gears and moving on to what one might do after taking a research job as a lab tech. I think one really good thing about taking a job as a lab tech is to use that as a way to decide if you actually want to go to grad school or not. You get to try out research without committing to the grad program. And that's actually one of the things that I was looking into when I took this job initially, thinking that I probably would want to go to grad school. And it actually helped me decide that I didn't want to go to grad school, which is great that I didn't end up getting into a program and ending up very unhappy or something like that and having to leave it early. Instead, I've just been working on similar skills, but without the stress of grad school. If you decide not to go to grad school and then do something like becoming a PI or becoming a professor, you also could get into higher ed administration if you like being in academia, but you don't actually want to be in a lab setting. You might also get into lab management. I know some people who have stayed in the system for a while and they're lab managers of bigger programs or programs that might have a lot of greenhouses, or they need some type of manager that manages all of the axillary pieces of their lab. You, of course, could take your lab skills and bring it to industry. Or, I mean, it's so diverse what you are able to experience in a lab setting. You could do anything. And I'm sure all the other career conversations have told you that you can go anywhere from where you are currently. And so this is my last slide. It's just some pros and cons I thought up in terms of this job and my experience in this job. So some of the pros, you have a lot of independence. If you like research and you're very curious, that can be really great. You often are able to get experience with lots of different techniques. I think I showed that. You're also working in a university, and so you have lots of resources. There's tons of seminars all the time that I'm able to go to. I also am allowed to take 4 credits per semester if I want to. So it's a great option if maybe you had I think they would let you do this, where like if you had a couple prereqs that you still needed to get done, but you also wanted to be doing research at the same time, you might be able to work it out with courses that you're able to take. I've also gotten to attend conferences, which are a fun experience. I have these in the middle because they kind of can go either way. So it's a really flexible schedule as a technician in terms of what I've found. But that could be a con because like if you're working with cell lines, you might have to go in on the weekends, things like that. Sometimes things are time sensitive, and so you might get to sleep in one day, but it might mean you have to work on the weekend or something like that. Also, I think in the middle is that the culture is very specific to each lab. And what I mean by that is each lab is, from my experience, is a very separate entity. And so like how your boss is dictates a lot in terms of what the culture in the lab is. And I think that's a little bit different than if you were in an industry setting, because in industry, there's levels of management who are involved. And in academia, it's really not that way. There might be a lab manager and a PI, so there might be 2 people, but it's really specific to each lab, and there's not much oversight between labs. Also, job security. So that's in the middle because your job usually is linked to grant money. But if you have a good PI who's good at getting grants, then they usually don't want to hire anyone new. So you usually can stay on as long as you want because they'll just build you into the grants. And then some cons. So for me, I think the fact that there's a lack of structure can sometimes be a challenge, especially when you're new to research, you don't have the experience to build the structure for yourself. So it definitely took a long time for me to feel comfortable in the lab because of that lack of structure. Also, it's a research setting and sometimes projects fail and you waste a lot of time, and that just can be hard to deal with. I spent the first 1.5 years on a failed project and nothing happened with it. So that happens. Also, there's high turnover in labs. People are constantly in and out. And then also as a lab technician, you are often the lowest person on the totem pole. And yeah, you can usually tell. Even if you're respected, you're not a grad student, you're not a postdoc, you're not a research associate. You are lowest man on the totem pole, lowest person. But that's where I will end this presentation. And then if people have questions, I definitely am happy to open up dialogue now. I don't actually, hopefully I didn't talk for too long.
Hi. Could you, just for those students who might not know, explain what a PI is?
Oh, yeah. Yeah, that's the primary investigator. So at big research institutions, there's professors, right? And professors are the ones who teach classes and things like that. But professors also have a research obligation where, and I think St. Mike's professors as well, it's just a smaller portion of their obligations. But they have to apply for grant money to fund their research programs. Mm-hmm. And they are on these grants, they are the primary investigators, and in their lab, they are the primary investigator. So they're the one that's driving the research and giving the direction to the research.
Great. All right. Well, that was very interesting. And I'm glad that you went through sort of what you did right after college and that you didn't jump right into a position. Because I think a lot of students are kind of scrambling right now. Things are still very competitive. Mm-hmm. And I think it's important for them to see that that just because you don't get a job right away doesn't mean you're never going to get a job, that the time will come. So. Right.
Well, and it definitely is true that being busy helps you be productive.
Yes.
And so even if you go and be busy while having fun, mm-hmm. you also are likely to be productive and maybe find what's next easier than if you were just,
I agree with that,
doing nothing.
All right. Hard. People can, if people have questions, feel free to unmute yourself and ask Brianne whatever you like. Don't be shy.
Any questions about anything?
I'm interested in going to research after graduation, and I guess my big worry is that I won't have the skills I need. So I was wondering if when you got your job, did you go through a lot of training or did they expect you to know everything right away?
That's a good question. I would say that they don't expect much of you. And if you're willing to ask questions, people are very much willing to give you information. I think it's also important to realize that every lab does things differently, and they know that every lab does things differently. So even if you are running, you've been in a lab, you know how to do DNA extraction and PCR. At your new lab, they might have a different protocol and they store their things in different locations. And there's just so many logistics when moving from lab to lab that I think if you're willing to ask questions, and hopefully there's one person that you are comfortable talking to, but there's not too much that's expected, especially if you feel like you have an awareness of some things, even if you don't know how to do them. If you understand some concepts, then it's okay.
One thing that I've come to realize after doing a number of these career conversations now and talking to other students who've gone on to get jobs, that what you say is absolutely true. The fact that you've done, gel electrophoresis a few times, you maybe couldn't do it completely on your own, but you've gone through that process. And as you said, labs will have different types of equipment and do things differently. Students in general have felt like they've been very well prepared for that first job. So I think it's important for you, Emily, to look at the courses that you've taken and make sure, pick out those skills that you've got. And all you're saying is that you've done this in a class, so you have a sense of what they're designed to accomplish, and you've gone through the process yourself. So just make sure you can identify those yourself and put those on your resume. I think that's important. But people will understand that you're a new graduate, and they'll understand the level of understanding of knowledge and experience that you have.
I did see a bunch of questions were popping up in the chat.
Oh, no, I haven't seen any. If you can see them, that's
I've seen a
good.
couple. Maybe they were just to me. Okay, so there's one here. Is it intimidating working independently among really smart individuals? I would say, yeah, I'm at Cornell, which has a reputation as being a very good school. There's definitely some intimidation there. And there's definitely some, how do I say this? Like, some attitudes of people are not my favorite, I would say, in terms of like people valuing work to a degree that I don't think is necessary, or things like that. There is some, especially grad students, there is a lot of pressure on grad students to work hard and to put in a lot of hours and things like that. And it would be easy to be sucked into that type of mentality working in the same labs. But I think one of the things that I had to do to make myself comfortable was to almost minimize how independent I felt. So there were a couple of times where I didn't have a direction for myself because I was new in the lab. I didn't necessarily know what to do next. And so I just invited myself to other people's projects, basically, knowing that if I'm working, especially because I had some free time. So like knowing that if I'm working with a postdoc, so someone who has a really developed project and they have a task for me, then that can put me into being not so independent. Hopefully that answers that question. Sorry, I'm just reading one that's asking.
For some reason, the chats, I can't see the chat, so you're
gonna. Yeah, they're direct messaging, so that's okay. Yeah. If I've ever wanted to be a PI of my own lab, and in a hypothetical where it required no grad school or anything like that, would it be something I was interested in? I do think that coming out of St. Mike's, I was definitely very interested in academia because I had such a great experience at St. Mike's. I think it's often, if you enjoy being a student, you might want to, like, you would enjoy being the professor, something like that. But I think I'm just not committed to the lab side of things, the research side of things. And so that has made me realize that the PI, the professor route, is not actually where I want to be. And there are just so many jobs out there that it's okay that that's not actually what I want to do. Like, there's lots of different paths that I could take. And once you start looking at jobs and things like that, you realize how much is actually out there. Okay. The next one, what advice do you have for undergrads trying to get into research with professors on campus? That's a good question. So I do think that one of the best ways that I was able to get connections with professors is by working really hard in their classes. So if you have a class that you enjoy or a professor that you really respect, or you're interested in similar topics, something like that, it does pay to make sure that you're present for their class if you're taking their class, and make sure you're engaged. And even if you're struggling in that class, being engaged and being the type of person that the professor is looking to, if they ask a question to the class, like now you're someone that they're thinking will answer, like having that type of relationship with the professor is really great. So then in the same way, if they come across an opportunity and you're the person that they look to in the class, now they might think of you when they are looking for a student to fill an opportunity that's fallen on their lap. So I definitely think that just being engaged in classes is the best way to make connections with professors. And then also definitely just reaching out to them, even if it's in person, that's great. If you're more comfortable sending an email, that's also good. And just tell them that you're interested in the topic that they're interested in as well. And you can ask them if they know of any opportunities for research. Again, usually people like making those connections. They like knowing where there's openings and they like knowing who would be a good fit. And if they know that you're looking for this opportunity, then it's much easier for them to give you the opportunity.
Just to follow up on that from the faculty perspective, and Peter and Carolyn might want to add something as well. You're absolutely right. There are students who are really engaged and interested. It doesn't mean they're always the best students in the class, but those are the students that you remember really. And they help make the class more interesting for everybody when they ask questions. So showing that level of interest is really important. And then, reaching out and asking if they know of any opportunities for research, even volunteering, saying, I have a few extra hours a week. Is there anything I can do in your lab or in your research project? Anything I can help you with? Just to get to know them a little bit better and allow them to get to know you a little bit better and see what kind of a worker you are, how you follow directions. All of those things are important because when we have you in class, we kind of know how you'll do on exams and papers and attendance and all of that. But sometimes it's important just to know, what do you like to be around when you're doing some work? And again, do you follow directions? Are you somebody who's responsible and will show up when they say they're going to show up? That kind of thing. So anything that can help a faculty member get to know you a little bit better, I think is really, really useful. And all of us are contacted pretty regularly with job opportunities. So it helps, as Brianne says, to sort of have some ideas about students that you might want to recommend.
Yeah, am I not muted?
You're not muted, Peter.
Okay, yeah, and I would add, you were talking, Brianne, about how important the reference letters are. And when you're engaged in class, it makes such a more connection than just getting an A on a test. And when you write a reference letter, you want to be able to, like in any good writing, if you remember from biocom, you want to be able to show instead of tell, kind of. And so you want to have specific examples backing up that they're engaged. And so I sometimes bring out little tiny points of something that happened in a class to illustrate how engaged the student was. And so I definitely reinforce the being engaged.
Yeah.
Okay, I have another question here. What general proportion of your job takes place in a lab doing research versus pure data entry, paperwork, meetings, etc.? I would say I'm maybe split 50-50 would be the best estimation. There are definitely depending on what I'm doing in the lab really dictates how long I'm in the lab. And like, there's definitely some days where I could spend all 8 hours of the day in a lab, at my bench. And then there's other days where I might be just doing a couple solution changes and really spend the majority of my day at a desk. In terms of meetings, I only have 2 hours of meetings a week that are definite. So I don't actually spend that much time in meetings, but they do pop up sometimes. I think that's all saying it. Here's a question. You said you might want to move up in the lab. Would you need to go to more school for this? How far can you move up in a lab without going to grad school? That's a great question. So like I said, I started as a lab tech 2, or sorry, lab tech 3 when I started the position. This past year, I was able to be promoted to a lab tech 4. And on the like HR scale of lab techs, it goes 1 to 5. So if I wanted to stay a lab tech, then I could move up to a lab tech 5. But you, like, if you want to really be having your own research projects and come up to the type of PI level, even if you don't want to have your own lab. So there's things like research associates that work in research institutions as well. But those people typically have a PhD. And so if you want to be in the higher level, I would say, in a research institution, you do need to have a PhD. I actually have some friends who have been lab technicians for a very long time, like 15 or 20 years, something like that. And, you know, they are experts in what they do, but they still didn't have the authority in the field because they didn't have that title. So like one person I know decided he was going to go to grad school because he just wanted that recognition, even though he already was an expert in what he does. But there are some hoops that academia needs you to jump through at some points in your career. And so, yeah, if you want to get high up in academia, then you would need to go to grad school. If you're out of academia, you don't need a PhD necessarily. Like if you're in industry, you don't necessarily need a PhD, but academia, you basically do.
Actually, in 2 weeks, the speaker who's going to be part of this series, Cynthia Turcott, graduated in '93, and she's in biotech industry. So she'll be a good person to talk to about what the research opportunities are if you decide to not go into academic research, but instead focus in biotech, biomedical kind of stuff.
It'll be a good comparison. Another question. Are you trying to gain new expertise and technical skills now in order to be prepared for your next job? Well, that would be very smart of me to do.
Just a suggestion, yeah.
Yeah. I would say that in my current position, I am definitely always open to new techniques.
Good.
And I do volunteer to do new things.
That's great. So have you made a decision yet about what your next step will be? Or are you still?
No.
No. All right, then we won't go there. I'll put you on the
but if anyone hears anything, shoot me an email.
Okay.
Another question in the chat was, did you ever feel stuck when graduating college and working jobs that you weren't using? That's another good question because I did do lots of jobs beforehand that didn't use my degree. And I would say that I did not feel stuck because I made the choice to do these other jobs. I could see how someone might feel stuck if they end up having to take a job that they don't want while they're waiting to look, or while they're applying to other jobs. But I would say that if you're ever feeling stuck at some point right after college, that right after college is a really great place to challenge yourself. And so if you've thought about moving somewhere or doing something completely different, that's a nice time to do that. So you don't always have to be stuck. And I know people have different resources, they have different life experiences. Like, it's not always an option to everyone to move or something like that. But there's other ways to try new things as well. So maybe maybe even if you end up back home and you end up working a job that you had worked during the summers or something like that, even if you're not able to get into a dream job right away, like try transferring to a new job or something like that. Just try changing up something if you start to feel stuck. It can be intimidating, but it usually works out for the better.
I think that's good advice. And because
Ooh, everyone
come
looks
away,
frozen. I'm
you-
wondering if there's some connection issues.
what happened?
All the student screens are looking very frozen, so I think something was going on for a second.
Is it? It may be close to 6. They may
Yeah.
have-
Well, things just reset. I think it was
okay.
something.
I was just going to say that sometimes students underestimate all of the different skills they come away from college with. We had speakers last week who were saying that although they're in the healthcare field, the things that really in some ways distinguish them among their peers is their writing ability. Interesting, which they felt St. Mike's really helped them with. And what was the other thing that they mentioned? They didn't say it, but I think I gathered that they were both very involved in like fire and rescue and move and that sort of thing. That interest in helping others, which is a very strong, theme at St. Michael's. I think that is something that is important too. It's really a skill, or at least an experience that some people undervalue themselves, but other people think highly of it. So take a close look at, all of the experiences and the skills that you're coming away with. And you're right, if you're feeling really stuck, then start looking around and seeing how else you can use the skills that you have.
Were there any other questions in the people on video? I thought I saw one person try to ask a question.
Maybe if you have any hands up, you can put their hands up electronically. I don't see any. So I think it's almost 7. So I think we can call it a night. And really, thank you so much, Brianne. I really enjoyed hearing you. And I know the students did as well. So thank you so much. And keep us posted as to what that next job is. Or if you change your mind and decide to go to grad school or whatever, just let us know.
Right. Or if I just, like I said, job security, if I just stay in
there you go.
biotech position.
Become an expert in something, which I'm sure you probably already are. Anyway,
thank you, everyone.
Very much, Brianne. Bye, everybody.
Yep.